Hey everybody, this is Carolyn Educator coming at you, and today's lesson is going to be on understanding the blood-brain barrier, better known as the BBB. So the reason why I am doing this video is because of pathological diseases such as dementia, Alzheimer's, and you do have caregivers out there that need help in understanding. And also there are essential oils that has the ability to cross the blood-brain barrier. And I just want to provide some information on what the blood-brain barrier is and its importance. And I've also included some video that can give you a little bit more detail than I can. So here we go. The blood-brain barrier is a highly specialized and protective barrier that separates the blood circulation from the brain's extracellular fluid, which is outside the brain. It plays a crucial role in maintaining a stable environment for proper neuronal function and protecting the brain from potentially harmful substances, which would be toxins. Anytime you see this word neuronal, we're talking about the nervous system, the neurons, the nerves, the synaptic transmissions, the electrical activity. The BBB is formed by a combination of endothelial cells, tight junctions, and various supporting cells, which we will get into those various supporting cells. So I'm going to try to do this. Okay, and it worked.
The blood-brain barrier, such as other blood barriers, is a remarkable anatomical adaptation. Its most striking structural feature consists in endothelial cells, tightly joined by tight and gap junctions. These structures bind the cells side by side through several proteins, including connexins, claudin 5, and ZO1, thus maintaining the barrier's integrity. Pericytes cover about a third of the capillary surface, and surrounding glial cells, named astrocytes, support these capillaries with their foot processes. This capillary bed in the human brain has a total length of approximately 600 kilometers, with a surface area of about 20 square meters. It is permeable only to some molecules, such as nutrients entering the brain, or waste products, hormones, and excess neurotransmitters that leave the brain. Only a small number of drugs are able to translocate it. To be able to do so, these molecules require lipophilicity, a size smaller than 500 Daltons, and evasion of mechanisms of active extrusion. Effectively deliver drugs across it. Specialists from around the world are called to discuss these questions on February 2004.
Next we have is the endothelial cells. So I just wanted to show y'all a little video. Endothelial cells is next. They are the innermost layer of blood vessels in the brain. It's lined with tightly packed endothelial cells. And I drew a schematic that I'm going to show y'all later on. These cells are connected by tight junctions, which are specialized protein complexes that create a physical barrier between the bloodstream and the brain tissue. So unlike other capillaries in the body, the endothelial cells of the BBB have fewer fenestrations, meaning small openings that make them less permeable. So I want to, let's see if I can get this right. Okay, let's see. Well, let me go full screen and get this ad. Okay.
The blood-brain barrier refers to the highly selective permeability of blood vessels within the central nervous system. The barrier controls, in a precise manner, substances that can enter or leave the nervous tissue. It helps maintain the stable state, or homeostasis, of brain tissue amid the fluctuations of circulating substances in the blood, many of which can act as neurotransmitters and could create chaos in neuronal activities if allowed to diffuse freely into the brain. The barrier also protects the brain from blood-borne pathogens and toxins. The blood-brain barrier is composed of several cell types, including endothelial cells that form the wall of blood vessels, Mural cells, namely pericytes, partially covering the outside of endothelial cells, and glial cells, astrocytes, whose extended processes, called end feet, wrap around the vessels. The endothelial cells alone can fulfill the functions of the blood-brain barrier, but their interactions with the adjacent cells seem to be required for its formation, maintenance, and regulation. The brain endothelial cells, unlike their counterparts in other tissues, possess unique properties that allow them to tightly control the passage of substances between the blood and brain. These properties can be classified into physical, transport, and metabolic categories. The brain endothelial cells are held together by tight junctions, which serve as physical barriers, preventing movements of substances through the space between cells. They have very low rates of vesicle-mediated transcellular transport. They control the movement of ions and substances with specific transporters, of which there are two major types: efflux transporters and nutrient transporters. Efflux transporters use cellular energy to move substances against their concentration gradient. These transporters are usually located on the blood side of endothelial cells. They transport lipophilic molecules, which have passively diffused through the cell membrane, back to the blood. Nutrient transporters, on the other hand, facilitate the movement of nutrients, such as glucose and essential amino acids, into the brain, down their concentration gradient. The brain endothelial cells also contain a number of enzymes that metabolize and thus inactivate certain neurotransmitters, drugs, and toxins, preventing them from entering the brain. An intact blood-brain barrier is critical for normal brain functions. Neurological diseases such as encephalitis, multiple sclerosis, brain traumas, Alzheimer's disease, epilepsy, strokes, and tumors can breach the barrier, and this in turn contributes to disease pathology and further progression. To note, however, that not all areas of the brain have the blood-brain barrier. For example, some brain structures are involved in hormonal control and require better access to systemic blood, so they can detect changes in circulating signals and respond accordingly. These non-barrier areas are located around the midline of the ventricular system and are known as circumventricular organs. Some of their bordering regions have a leaky barrier. The blood-brain barrier also has its downside. While it protects the brain from unwanted drugs and toxins, it also prevents therapeutic drugs from entering the central nervous system to treat diseases. Several strategies are developed to overcome this obstacle, including delivering the drug directly into the cerebrospinal fluid, use of vasoactive compounds, designing drugs with higher lipid solubility, hacking the endogenous transport system to carry the drug, and blocking the efflux transporter that pumps the drug back to the bloodstream.
All right, now that we watched the part on the endothelial cells, let's talk about the function of the blood-brain barrier. The primary function of the blood-brain barrier is to protect the brain from potentially harmful substances. So I mentioned that before, including toxins and pathogens. So let's go over pathogens. That would be your microorganisms, which include bacteria, viruses, fungi, while allowing the passage of essential nutrients, oxygen, and other molecules necessary for normal brain function. The selective permeability of the blood-brain barrier is a critical side of maintaining homeostasis within the central nervous system. Now we have tight junctions. Tight junctions are protein structures that seal the gaps between the adjacent endothelial cells, the ones that are near. And I have a video for that. They prevent the free movement of molecules between the cells, creating a tight barrier that restricts the passage of substances from the blood into the brain. This selective permeability, meaning semi-permeable, is essential for maintaining optimal chemical environment required for proper, and there go that word again, neuronal function. So I also have a video for you to watch. Let's see if I can get it to play this time. This one not playing, y'all know what I'm doing wrong. Nope. Let's see. It worked before, so I must be doing something. All right, let's do it this way. Nope. All right, we'll do it this way. I'm gonna copy it. And we're gonna put it here. And I'm gonna stick it in there.
Here's one thing you need to do before buying
with these
anything online. Don't
commercials.
spend another dime on Amazon until you watch this first.
Sometimes.
To keep out foreign invaders, the brain relies on a membrane that lines the brain's blood vessels, known as the blood-brain barrier. All blood vessels are lined with endothelial cells. There are gaps to let molecules out of the blood vessel and into other tissues. But the endothelial cells that line the brain's blood vessels are really close together. That means only certain molecules can slip through and get out into the brain tissue. Some molecules, like water and oxygen, can cross the blood vessel's wall and get into the brain. So can some fat-soluble substances like caffeine and alcohol. Other molecules the brain needs, like glucose, can hitch a ride into the brain tissue with the help of certain proteins. But by and large, the blood-brain barrier doesn't let most drugs through, which presents a big problem in medicine. That's made it tricky to find treatments that work well for neurological conditions like Alzheimer's, stroke, and brain cancer.
Okay.
Dementia is a general term for a decline in memory and other cognitive abilities. It is not a disease on its own, but rather a group of symptoms caused by an underlying condition. Most dementias worsen over time and are irreversible, but some types can be reversed with treatment. While the incidence of dementia increases with age, it is not a normal part of aging. The most common cause of dementia, responsible for more than 50% of all cases, is Alzheimer's disease. In this condition, abnormal toxic deposits of proteins, known as plaques and tangles, cause the death of neurons. The damage initially takes place in the hippocampus, the part of the brain that is essential in forming memories. Short-term memory loss is usually one of the earliest symptoms. Most patients show first signs of mental decline after the age of 65, but for a small subset of cases, the disease runs in families and strikes earlier in life. Second to Alzheimer's is vascular dementia, a condition in which poor blood supply to the brain impairs normal function of neurons. Symptoms may appear suddenly after a stroke, in a stepwise fashion after a series of mini strokes, or gradually as a result of age-related vascular wear and tear, or any conditions that damage or narrow blood vessels over time, such as high blood pressure, high cholesterol, and diabetes. Incidence of vascular dementia increases with age and cardiovascular risk factors. In the third place is Lewy body dementia. Lewy bodies refer to abnormal protein clumps typically found in neurons of these patients. The earliest and also most prominent feature of this type is a sleep behavior disorder in which patients physically, sometimes violently, act out their dreams. Other early symptoms may include visual hallucinations. Memory loss may not be noticeable until later stages. Dementia caused by advanced Parkinson's disease belongs to this group. Frontotemporal dementia is another common type of progressive dementia. This group is characterized by neuronal cell death in the frontal and temporal lobes of the brain, the areas associated with behaviors and language. Common signs and symptoms include changes in behaviors, apathy, blunting of emotions, and language deficits. A significant portion of this type has a strong genetic component and tends to occur early in the middle-aged population. More than one type of the above-mentioned dementias may coexist in one patient. Less common causes of dementia include Huntington's disease, Creutzfeldt-Jakob disease, and traumatic brain injuries. Dementia may also develop as a result of endocrine or metabolic problems, such as thyroid disorders and vitamin deficiencies, or infections such as Lyme disease and neurosyphilis. For these types, symptoms can be reversed with treatment of the underlying condition. What's the difference between Alzheimer's disease and dementia? Perhaps you've been confused by these terms in the past, or mistakenly thought they were the same thing. Well, you wouldn't be alone, because knowing the difference between Alzheimer's disease and dementia can be tricky, but it's actually not too hard to understand. In a nutshell, it's like the difference between shortness of breath and asthma. Shortness of breath is not a specific disease. It can have many different causes, and asthma is just one of them. Likewise, dementia is not one specific disease. It's a combination of symptoms that has many different causes, including Alzheimer's disease. Still sound a bit confusing? Okay, think of it like this. You go to your doctor with shortness of breath. To find the cause, she listens to your chest to see if it's due to asthma or pneumonia. And then she checks your heart to see if your breathlessness is caused by heart failure or a very rapid heartbeat. She even asks if you've been worried in case your breathing difficulty is anxiety related. After her examinations, she concludes that your shortness of breath is actually a nut allergy. Similarly, when dealing with dementia, your doctor needs to figure out the underlying cause. And while Alzheimer's disease is the most common cause of dementia, it's not the only one. Dementia can also be caused by vascular changes in the brain, Parkinson's disease, Lewy body disease, vitamin deficiencies, thyroid gland problems, chronic brain infections, or medications. Even a very deep depression can sometimes look like dementia, which means if someone you love has dementia, it doesn't necessarily mean that they have Alzheimer's disease. And that's very important for your doctor to establish, because some causes of dementia, such as infection, medication reactions, an underactive thyroid gland, or vitamin deficiencies, can be reversible with the right treatment. So figuring out the cause and a plan of action could improve your quality of life. And that's why, if you or your loved one have the symptoms of dementia, you need to see your doctor to find out the underlying cause.
So I showed you some videos, and I wanted you to see those two videos on dementia and Alzheimer's. Also showed you a video on tight junctions. So now we're on parasites. Parasites are cells embedded in the basement membrane that wrap around capillaries. They're thought to contribute to the regulation of blood flow and the development of the blood-brain barrier. Interactions between the parasites and endothelial cells influence the tightness of the barrier. Then we have astrocytes. Astrocytes are star-shaped glial cells that play a crucial role in maintaining the blood-brain barrier. They extend their processes to envelope blood vessels and release chemical signals that regulate the tightness of the endothelial junctions. Astrocytes also contribute to the induction and maintenance of the blood-brain barrier. So I want to show you a video next.
A human brain contains billions of neurons. Neurons are probably the more important and better known cells of the brain as they carry out the brain's communication function. Less known are some trillions of support cells called glia or glial cells. The glia may not be the center of attention, but without them, neuron functions would be impossible. The major types of glial cells in the brain include oligodendrocytes, microglia, and astrocytes. Oligodendrocytes are specialized cells with long processes that wrap around axons of neurons to form the myelin sheath. The myelin sheath acts like an electrical insulator around a wire. It helps to speed up the electrical signals that travel down an axon. Without oligodendrocytes, an action potential would propagate 30 times slower. Microglia are special macrophages found only in the central nervous system. They wander through the brain tissue, phagocytizing dead, injured cells and foreign invaders. High concentrations of microglia are an indication of infection, trauma, or stroke. Astrocytes are the most abundant and functionally diverse glia. These star-shaped glial cells provide supportive frameworks to hold neurons in place. They provide neurons with nutrients such as lactate. They also produce growth factors that promote neuron growth and synapse formation. It's been suggested that astrocytes can control how a neuron is built by directing where to make synapses or dendrites. Through their processes, known as perivascular feed, astrocytes induce endothelial cells of blood vessels to form tight junctions. These tight junctions are the basis of the blood-brain barrier that restricts the passage of certain substances from the bloodstream to the brain tissue. Astrocytes help to maintain the chemical composition of the extracellular fluid. They express membrane transporters for several neurotransmitters such as glutamate, ATP, and GABA, and help to remove them from synaptic spaces. Astrocytes also absorb potassium ions released by neurons at synapses. This helps to regulate potassium concentrations in the extracellular space. Abnormal accumulation of extracellular potassium is known to cause epileptic neuronal activity. Recently, it has been shown that astrocytes can also communicate electrically with neurons and modify the signals they send and receive. In a manner similar to neurons, astrocytes can release transmitters, called gliotransmitters, upon stimulation. This suggests that astrocytes may be involved directly in the communication functions of the brain. From the clinical viewpoint, neurons have little capacity for renewal and therefore rarely form tumors. On the contrary, glial cells are capable of dividing throughout life and are the primary source of brain tumors. Thank you for watching.
So you've seen a video on astrocytes, tight junctions, and also what the blood-brain barrier is. So finally, I want to share this. While the blood-brain barrier is essential to protecting the brain, it can pose challenges for the delivery of drugs, meaning medications, to the brain for the treatment of neurological disorders, any type of nervous system diseases. Researchers are exploring various strategies such as nanotechnology and targeted drug delivery systems to overcome the limitations imposed by the blood-brain barrier and enhance the delivery of therapeutic agents. However, there is something, I don't have it with me. There's something that I wanted to share with you in reference to essential oils and the blood-brain barrier. And lastly, I want to share with you scientific research. This is, to me, this may be an ongoing research. The potential effects of serotonerpenes, such as those found in frankincense essential oil, on dementia or Alzheimer's disease are areas of interest in scientific research. This is always going to be ongoing. There are doctors, there are scientists, and there are researchers that have been actively doing research on this, as well as doing research on gathering data and actually putting it into physical theory, meaning putting it into use on individuals, meaning the essential oils, and using the essential oils on people for the effects of the, so I could never say this word, serotonerpenes, the effective use of slowing it down because these terpenes, meaning these sequet terpenes, have neuroprotective properties. So that's what makes it very important, because they do have neuroprotective properties. So this is, something that I wanted to share. I hope that you have gotten some benefit out of it, that you have learned something from it. And I will see y'all into the next class. Take care.